Okay, we've probably done the most important thing for replication, which is to understand the mechanism of it. But there's still some uh, context, particularly biological context, that we want to put this into. Let's go on to the next page and let's set up a replication bubble again. Remember, DNA is pulled apart, but the strands are still anti-parallel. We said previously that when we have a bubble, we start with primers. I'll make those red for RNA. And we extend DNA off of the 3' ends in this fashion. So this is new DNA here. And these are RNA primers. Now, what we can say is that DNA can continue to be replicated, and it can go on basically forever till we get to the end of the chromosome where we run into a new bubble. We can do the same up here, and we can continue on going. We do have a problem though, and that is, how do we replicate this area up here and this area down here? And the big problem arises because this part of the RNA is the 5' terminus, and that's true for this as well. And what we said on a previous video is that you can only extend DNA off of the 3' end. So we cannot draw an arrowhead on the 5' end and continue on in that direction. Here's how it happens. What we're going to do is actually put in another primer in these positions. Really, those are on the outside of the bubble. And what we can say is this is the 5' end, and this is the 3' end. Same thing over here. We've got the 5' end and the 3' end of these new primers. And we can then use DNA nucleotides to backfill. And this is a different type of replication, or at least it's a different type of fragment that's being synthesized. And what we call these new smaller fragments that come from the outside of the bubble to the inside are lagging strands. There's a reason for that, I'll tell you in just a minute. This is a lagging strand over here as well. These are also named sometimes after the person who discovered them, which is a Japanese scientist. They're called Okazaki fragments. Now, the other strands that we talked about earlier, the ones that can continue on going without any problem, they're going from the inside of the bubble to the outside toward the end of the chromosome. These are going to be called leading strands. Now, the addition of Okazaki fragments allows us to get all of the area replicated, not just this one and this one, but now we can replicate these as well. But it leaves some problems. First of all, we have some RNA that's still present. In fact, we got a lot of RNA. We got 4 primers in red shown there that are still present, and we've got to get rid of those. We also have gaps, and the gaps are seen like here. We can bring that DNA right up to the RNA, but we can't seal them together. Those are problems that are going to all have to be handled. But let's just finish this Okazaki fragment video by pointing out that as we continue to expand the bubble, we're going to have to put in more primers and do more backfilling like this. And we keep making these small Okazaki fragments. They're, 500 to 1,000 bases long. And on the other hand, we can take our leading strands and they just continue on. There's no need to continually put down new primers. This one primer will serve as we go in that direction. So the upshot, let's finish this off by making a note. Okazaki fragments is the name of this topic. And these are necessary because all DNA replication or all DNA synthesis must extend off the 3' hydroxyls. So all of the DNA synthesis we have, whether it's leading strand or lagging strand, is proceeding in the 5' to 3' direction, and that allows us to get all of it copied, at least into RNA and DNA. We'll take care of the other problems I mentioned on the next video.